Hello, everyone. I hope this finds you well. This is our second week of Q4, the fourth quarter, and I want to begin it on a good note by making sure I personally went through the slides with you. I wasn't able to do it last week. So the topic for this week is going to be DNA and proteins. And I'm just going to go through not every slide, because at the end we're going to have some practice slides and show you how fun going through yourself. I just kind of wanted to talk us through the slides and some of the topics that we're going to be that are going to be the topics of what you're going to be doing this week. Okay, last week we talked about DNA, and basically we said that it was it's the blueprint or the instructions for the cell, and each cell reads the DNA and then produces proteins, and the cell uses the proteins to perform each of its functions. And this slide just shows you a variety of the different types of functions in the body. The big picture, genes and DNA, and the DNA basically code for proteins. You need to understand that your actual protein sequence, that's what gives you the specific traits you have, like your skin color, eye color, etc. And the proteins are made in each cell, and they determine the way the cells look and their actual individual function. Okay, gene regulation. Every cell in an organism has the exact same DNA, but each cell only turns on the genes it needs to function. And here's just an example of the difference between human skin tissue and your cardiac muscle cells. Okay, the genes turn the heart cells on to form muscle cells, as opposed to what skin cells that would be needed. Also, this is kind of interesting. You have special skin cells that make up your skin pigment. Eumelanin is darker pigment, and pheomelanin is a lighter pigment. And this is one of the things that specifically proteins are given like code names, like MCR1 protein, which is a type of melanin that's produced by the skin. The MCR1 gene is instructions for how to make the MCR1 protein. Okay, and this is it, like I said, in terms of skin tone, skin color, this is the particular gene that pertains to that. Now, as you can see, that's basically the different sequence, and it's so important. By this point, you should at least have, and we're going to talk about it in a few minutes, but you should at least have the base pairings down. It's very important that we have the base pairings for DNA down. Guanine pairs with cytosine, and adenine pairs with thymine. It's so important because going forward, if you do not understand that, and if you please email me or let me know, it's going to be a little bit harder to understand when we talk about RNA. But different genes make different proteins, and especially in terms of skin color. Okay, so changing the gene, if you change the gene, that changes the protein. If you change since amino acids are the building blocks of proteins, if you change the amino acid chain, you change the protein, and thus you change the trait. That's very important because it's they're unscheduled changes that we're going to talk about, which are called mutations. It's important that you're going to make that connection between unscheduled changes in the sequence, which that when the protein changes, sometimes it can change change things in a bad way. Also, all cells contain the same DNA and the same genes. Different body cells only turn on the genes that to make the proteins to do certain jobs. All right, I'm moving right along. Not to keep you too long here, I just wanted to hit the highlights. How does protein synthesis work? Now, of all the slides, this is like the best slide because this ties it all in together. Because we have to understand that we start with DNA, and DNA actually starts in the nucleus. Okay, your DNA is in the nucleus, but once the purpose of RNA is you had to have something to go in the nucleus and make a copy of the DNA, because the DNA only stays in the nucleus, and the DNA is your true genetic code. So, and then once you get the RNA takes the genetic code out of the nucleus from the DNA and goes to the ribosomes and assembles chains of amino acids. Now, the amino acid chains, I'm sure you heard before, amino acids are the building blocks of protein. The specific amino acid chain sequence codes for proteins, and those proteins make the actual traits. That's what gives you your eye color. And that's what gives you your skin color and everything like that. But it's important to make the connection between your traits and DNA. Okay, the original DNA has to stay in the nucleus. That's what's in your chromosomes. Okay, so you had to have something or some way to copy that and get it to leave the nucleus and actually code, to carry the genetic code to actually assemble amino acids and then proteins. Okay, I was talking about this before, very important. The DNA versus RNA is very important that you see, you could get the difference between DNA and RNA. And the difference between DNA and RNA, the main difference is that one of the nitrogenous bases, there's four. One of the nitrogenous bases for RNA, okay, is different. RNA has uracil instead of thymine. So what does this mean? What that means is that thymine would normally pair up with adenine, but instead of putting a T when you see adenine to pair up with adenine, whenever you see adenine, you're going to pair it up with uracil. Okay, and that's the only thing that's different. Okay, in terms of the nitrogenous bases. The other difference in RNA is where the simple sugar, because we know the components of a nucleotide, okay, involved in nucleic acid is the simple sugar, a phosphate, and a nitrogenous basis. Well, the simple sugar for DNA is deoxyribose, and the simple sugar for RNA is ribose. Okay, it's important, folks, for you to kind of know this code, okay, because it's pretty much the only way you're really going to be able to understand protein synthesis and exactly how the amino acids are assembled. Okay, another difference, and this is a big difference between DNA and RNA is obviously, as you can see, RNA is single-stranded and DNA has a double helix or a double strand. So like if you had one strand on top of each other for DNA, the top strand it has the bottom strand is a complementary strand of this top strand. Whereas for RNA, since RNA just has to go copy one of the strands of DNA, it's only single-stranded. Okay, and again, RNA has cytosine, guanine, adenine, and uracil for its nitrogenous bases, and the DNA has cytosine, guanine, adenine, and thymine for its nitrogenous bases. Okay, how are proteins made? Now, I'm sure you've heard that amino acids are the building blocks of protein. But the way they actually build proteins is that strings of amino acids together, okay, make up a protein. So the transcription, it takes place when RNA goes in and makes a copy of the DNA. That's transcription. What I do when I transcribe something, I'm copying something from one thing to another. Translation occurs when at the ribosome, when the DNA code is coded into actual amino acids. And this is the interesting part, and I always felt it was kind of cool part, because this is the place where biology and chemistry meet. Because in a laboratory, you can actually make the amino acids in the laboratory. So conceivably, you could actually artificially do protein sequences and also make a genetic code. So if you just make up, like a lot of times we just make up just willy-nilly a DNA sequence, that is actually somebody's genetic code somewhere. Okay, how does a cell know how to put the protein together? Okay, all of the instructions are in DNA, and one gene can be used to make one protein. And that's what I was mentioning. Transcription is when RNA goes in and makes a copy, as you see right here in the nucleus. Why? Because once again, the DNA never leaves the nucleus. Okay, and translation is when on the ribosome that the RNA code actually codes for specific amino acids. And it's those chains of amino acids that actually make proteins and protein sequence. So transcription, and there's actually a lot of steps. The actual DNA double helix has to unwind and allow the RNA that comes in there. And the actual translation that comes in is when the RNA goes back out of the nucleus into the cytoplasm on the side of the ribosome and actually assembles the amino acid chain. Okay, the cell reads for transcription, the cell reads the gene from one strand of DNA. Kind of what I'll let you guys get into that. This is good practice on seeing what the what the transcription takes place and exactly how the RNA molecule is formed based on the DNA strand. Okay, and I'll let you guys. This is basically what I was been talking about. Translations is when the actual amino acids are assembled at the ribosome. Now, here's interesting because, and you'll be able to look at a chart when you practice. They're going to practice with you using these codons. Now, these three letters actually code for actual amino acids, and you'll be able to assemble them. But I'm going to allow you guys to have fun doing that. So once again, translation is when the genetic code that was copied in RNA is actually codes for actual real amino acids, the building blocks of protein, and the amino acid chains on the ribosomes are actually assembled. So to kind of recap, the main idea is protein synthesis is a process of making a protein. Transcription is when the DNA sequence is copied by RNA. Translation is when the RNA in three-letter codons actually codes for the assembly of the amino acid chain. Okay. Now, I'm going to stop here. I'm going to shut it down here. I want you to go through these practice items because it will definitely help you understand. If you have any questions, we will be having a meeting, our office hours. I would go ahead and dial into the office hours that I'm going to post and make sure you ask questions, okay? All right, thank you very much, and I hope you guys have a great day. Hope this was informative for you. And I'll talk to you next time.